For decades, the standard medical curriculum has taught osteoarthritis as an entirely mechanical problem. The model is highly intuitive. Joints are biological hinges. Cartilage acts as a shock absorber, and over a lifetime of weight-bearing movement, that cartilage simply wears away from physical friction. But clinical observation consistently breaks this model. Physicians frequently document severe joint degradation in patients who have no history of high-impact athletics, acute trauma, or excessive physical load. This discrepancy has redirected research focus toward the underlying biochemical environment of the joint. To understand why cartilage fails, we have to look past the physics of weight and movement and examine the chemistry of the tissue itself. Treating osteoarthritis solely as a mechanical defect addresses the late-stage symptom while leaving the biological drivers of the deterioration unaddressed. That biological perspective is the focus of an emerging discipline known as metabolic rheumatology. In this framework, mechanical friction is a secondary factor. The primary variable driving cartilage destruction is the systemic concentration of blood glucose. This model tracks how free glucose molecules physically infiltrate the dense, fibrous extracellular matrix that surrounds the joints. Once inside, high concentrations of sugar execute two specific modes of destruction: the structural rigidification of the tissue and the triggering of targeted enzymatic destruction. Systemic glycemic load operates as the silent architect of joint failure. The first vector of attack relies on a biochemical process called in-vivo glycation. This is a spontaneous, non-enzymatic chemical reaction where free-floating sugar molecules bind directly to structural proteins in the body. It is the same chemical mechanism as the Maillard reaction, the process that causes sugars to brown and caramelize when cooking. Within living human tissue, this process occurs continuously at body temperature. When the glucose collides with a protein, it permanently fuses via a covalent bond, transforming the smooth protein chain into a dense, deformed structure. In clinical terminology, these malformed structures are classified as Advanced Glycation End-products, or AGEs. Once this chemical bond occurs, it is irreversible. The body lacks a physiological mechanism to break these covalent bonds apart. Because they cannot be cleared, AGEs accumulate over a lifespan. However, chronically high blood sugar accelerates this process, multiplying these microscopic collisions. A sustained high glycemic load continuously drives the conversion of healthy tissue into crystallized, compromised matter. To understand how this crystallization destroys a joint, we have to examine the anatomy of healthy cartilage. The extracellular matrix is a lattice built of collagen fibers for structural support and proteoglycans, which retain water to provide elasticity and shock absorption. As excess blood glucose permeates this matrix, advanced glycation end-products begin to form directly on the collagen fibers. The AGEs undergo a process called covalent cross-linking. They create rigid chemical bridges between independent collagen fibers, locking the lattice tightly into place. Tissue that evolved to be supple and dynamic becomes rigid, reducing its ability to absorb mechanical impact without fracturing. This physical stiffening is only part of the failure. The glycation process also impairs the chondrocytes, the specialized cells responsible for maintaining the cartilage. As these cells are affected by glycation, their ability to synthesize new proteoglycans diminishes. Without proteoglycans, the matrix cannot retain hydration, and water molecules dissipate. This loss of hydration strips the tensile strength from the joint, leaving the tissue vulnerable to tearing under normal mechanical stress. Systemic sugar dehydrates the cartilage and stiffens the matrix, turning a biological shock absorber into a brittle structure. This mechanical failure is only half of the pathology. The presence of glycated proteins also triggers a localized immune response. Joint cells possess specific surface proteins known as RAGE receptors. That stands for Receptor for Advanced Glycation End-products. These receptors act as biological tripwires. When an accumulated AGE molecule makes contact with the cell membrane, it locks into place on the RAGE receptor. This binding event triggers an intracellular signaling cascade, sending an alert deep into the nucleus of the chondrocyte. The cell responds with a release of inflammatory cytokines, flooding the immediate joint space with chemical signals. This localized cytokine storm stimulates the production of a specific class of enzymes, matrix metalloproteinases. Metalloproteinases act as a cellular demolition crew. Their function is to chemically digest and dissolve whatever healthy collagen remains in the extracellular matrix. By saturating the joint with AGEs, systemic sugar leads the immune system to actively break down its own cartilage. This flowchart summarizes the dual-vector nature of metabolic osteoarthritis. A high glycemic load simultaneously drives mechanical cross-linking and RAGE-mediated enzymatic destruction. Sugar provides the material that rigidifies the joint and the trigger commanding the immune system to digest it. These vectors create a self-sustaining feedback loop. The mechanical brittleness causes microtears, generating local inflammation, which further accelerates enzymatic breakdown of the tissue. Treating a degraded joint through physical therapy or surgery, while leaving this systemic metabolic loop unaddressed, leaves the biological driver of the disease in place. This biochemical evidence necessitates a different approach to musculoskeletal longevity. Regulating a patient's systemic insulin levels and controlling their glycemic index is not only a protocol for managing diabetes or obesity. Glycemic control is a primary tool for preserving the structural integrity of the joints and managing chronic pain. The long-term consumption of simple sugars extracts a physical toll on the human skeletal system. To truly protect the complex mechanical machinery of the human body, we must first secure its underlying metabolic foundation.